What's up, guys? Another episode of 461. We're portable today. Don't have access to a whiteboard, so we're going to be doing it on the iPad. Okay, so today we're talking about ventilation. We're talking about negative pressure. We're talking about the lungs, different things that can happen there, oxygen, carbon dioxide, etc. We're going to start by talking about negative pressure, how the lungs kind of work, problems that can happen. So here we have a drawing of the lungs. Let me take that off. So here we have a drawing of the lungs. Now, as you can see, this blue, that's our bronchi, and then it's going to branch into your orange, which is your bronchioles, and then those are going to branch, those green dots are all alveoli. Alveoli is where gas exchange is actually going to take place, and you have different sized alveoli. Around your lung, that black line's your lung, and then the purple is what's known as your visceral pleura. It's a very thin membrane that covers the lung, and then you have a space called the pleural cavity filled with fluid called serous fluid, and then you have that blue line all the way around, which is called your parietal pleura, and that's going to coat the edge of the thoracic edge of your thoracic cavity. Okay, so basically what happens when your lung collapses, or not collapses, that's a bad word for it, when it shrinks, when you're breathing out, what happens is this gets smaller. Okay, so it goes like that, and this purple is going to pull on the visceral pleura is going to pull on your parietal pleura. Think of it like this. Okay, if I start to pull out, I'm going to create a negative pressure here in between my two hands. Same thing happens. You're going to create a negative pressure there in that pleural cavity, and that is going to pull the lung kind of out, cause it to contract a little bit, which is why your lung doesn't actually collapse when you breathe out. Your lung wants to collapse, but that negative pressure is actually protecting it. So say surfactant. Okay, so surfactant, it's made up of a hydrophilic phospholipid as well as a hydrophobic protein. Basically, what's going to happen is your surfactant is going to make it so that your surface tension gets distributed evenly among all the alveoli. Like I said before, we have different sized alveoli. So if you didn't have surfactant, what would happen is you have something called Laplace's law. Laplace's law states that as you increase in surface area, you decrease in pressure. Okay, so that means that bigger alveoli require less pressure to inflate, and smaller alveoli require more pressure to inflate. So what does that mean? We know that air wants to go from high pressure to low pressure. So that means that as you try and inflate the lung, you would cause the high pressure, the air in the high pressure, the small alveoli, to go into the low pressure, the big alveoli, which could overinflate the alveoli and actually not cause the small alveoli to inflate at all. So what surfactant's going to do is surfactant's going to coat your, it's going to coat, I believe, the visceral pleura, not the visceral pleura, the alveoli, and it's going to equalize the surface tension because surfactant's going to collect more on the small alveoli, and it's going to make it so that the surface tension is equalized among all the alveoli, which makes it possible to actually just breathe normally. Okay, now we're going to talk about pneumothorax. So pneumothorax, it occurs when you get air inside of this pleural cavity. Can happen. You can get shot. You can have a collapsed lung. Basically, what happens is you have air in here, which makes it so that you're basically, you have too much air in there. It's compressing your lung. You can't breathe. So what happens is we insert a knife, a needle. It depends. We'd like to do just a needle, but if you're on the fly, a knife works. So you'll do it into the thoracic region, in between the ribs, just until you puncture this pleural cavity, and then that'll allow the air to escape, allow your lung to actually reinflate. Okay, now we're going to talk about the alveolar gas equation. So the alveolar gas equation is, PaO2, that's measuring your oxygen that's actually in the alveolus. So it's PaO2. If we want to find that, we take our Fi, which is our fraction of inspired O2, how much, breathing in, and we're going to times that by your atmospheric pressure minus your H2O. So as oxygen moves down the lungs, it's actually going to get diluted by water. So that's that over there. So ATM's about 760, H2O is usually about 50. So what happens is we solve for that, and we take our FiO2, our fraction of inspired oxygen, is usually about 0.21. So that's going to give us a PaO2 of usually about 100, which is normal. If it's too high, too low, we can have problems. So we're going to get a PaO2 of about 100. Now, if we know this, we know that this is all the oxygen in the alveolus, which means the oxygen in the blood should be about the same, should be about 100. If our PaO2 is a lot bigger than the oxygen in the actual blood vessel down here, that means there's a problem with transferring stuff. Something is stopping oxygen from going into the blood. We need to figure out what it is, or there could be consequences. Okay, perfect. Now we're going to talk about transportation. So basically what happens here, same thing. So you have your, all this is oxygen, all this orange stuff. So you have your oxygen here in the alveoli. It's going to diffuse out here into the blood. And from there, it's going to come onto a red blood cell. Okay, this big thing right here. Here you have your hemoglobin groups, those proteins on the blood. You can have 4 oxygen per hemoglobin. Okay, 4 oxygen per hemoglobin. And basically what's going to happen is it's going to bind to it, and then that's going to create a gradient. So what happens is, remember, this is about 100. So let's say 4 leave over here. So now this is 96, this is 104. Well, you have less oxygen here in your red blood cell, so oxygen's going to come down into the red blood cell. That's going to create this back to 100, and then more oxygen, or this is going to drop down, and so more is going to come over here to kind of equalize it out. So you just have a constant transferring, kind of like just going down, kind of like a waterfall, cascade sort of thing. So basically then what happens is it comes down here. You get down to the tissue where your oxygen's going to come off the hemoglobin, and the same thing, cascade. So you have a higher concentration here, it's going to come off, and then from there in the blood, it's going to go into the tissue cell. Then it's going to replenish and go the other way. At the same time, you have CO2 from the tissue going to jump into the blood, going to bind your hemoglobin. Hemoglobin comes back up, CO2 hops off, and then it's going to go into the alveoli, into the lungs, and then we're actually going to exhale our CO2. Okay? So now we're going to talk about the oxygen hemoglobin dissociation curve. Basically, what happens here, remember, part of your hemoglobin is your heme group. We talked about that last unit in cardiology. And your heme group carries the oxygen. Basically, what's going to happen, you have your iron. So if you're deoxygenated, as you can see right here, your heme is actually going to be domed, which makes it so that oxygen can't come here. Okay, so the doming of that is actually caused, it's actually called the T state because it's tense, and then the regular one, oxygenated, is called the R state. And basically this can be caused, it's caused by something, it's called 2,3, I believe, wait one second, let me find it. 2,3-BPG, 2,3-bisphosphoglycerate. So basically what that happens is that's going to take and shift the phosphates over 1, and then to do that, that's going to cause it to get domed like that. So now talking about the oxygen hemoglobin dissociation curve. As you can see, so this is normal. So this is your O2 saturation, and then this is your PO2. So you want it to kind of look like that. See, oxygen released, oxygen released. Now, if you have over here this right shift, means we're more likely to be in our T state. Remember, that's our tense state. That's where we have our 2,3-BPG. That we don't want that because that makes it a lot harder to get oxygen saturation, which is why that doesn't go as high. It comes that way a lot faster. We would like to be regular, and then over here after 2,3-BPG is about to drop rapidly. We have a left shift over here, which means we're more likely to be in the R state, which means that we have a ton of oxygen, really saturating the things. Okay, so as you can see over here, kind of how those are all affected. If we affect this, if we make it so that we can't get oxygen, it makes it so that our hemoglobin can't get saturated, which actually causes a right shift in our oxygen hemoglobin dissociation curve. We want to be normal. We don't want to be in a right shift, because if we're in a right shift, that means we can't saturate with oxygen, And as you can imagine, that can have all sorts of trouble clinically. So thanks so much for watching, guys. Have a good one.